Hello, everyone, and welcome back. It's time to start our event today with our first speaker. Our first speaker, as you could appreciate from the banner in the short interlude, will be Gerrit Borchard, Professor Gerrit Borchard. He will be giving the introductory words today. Professor Borchard is a professor of biopharmaceutics at the University of Geneva, and he will be speaking today some words about academic and industry liaisons. So without further hesitation, I would like to welcome Professor Borchard onto the stage.
Yes, thank you. Can you hear me? Can you see me?
Yes, we can hear and see you very well. Good morning,
Okay.
Gerrit.
Good morning. How are you?
We're doing good. We're doing good. You?
Yeah. Excited to be given the opportunity to present here today. Thank you.
Great. We're also very, very happy that you want to do the opening words today. I see you have prepared a beautiful presentation for us.
Let's wait and see.
I can't wait to hear it. So without further hesitation, Gerrit, the floor is yours.
Thank you very much. Let's put this into presentation mode. I can't see you anymore, but I think that's okay. So good morning to everybody. And again, thanks for giving me the opportunity to talk a little bit about my way between academic and industrial research. I picked this image here of Midtown Manhattan, not because New York City is one of my favorite towns, but because it also shows you a dichotomy between the highly ordered structures here of the avenues and the streets and this more chaotic part where you have meandering paths here, which sometimes end nowhere. Now, for me, this is an image for two things. One thing is, this is what you put in your CV, right? This is what you say, okay, I did this and then I did this in order to impress your future employer. However, the green part here, the more chaotic one, is how it actually happened, the story behind this. You can also look at this as a thesis. Now, the structured here is more the outcomes or the thesis. You go from hypothesis, you do some experiments, in the end you have some results, very straightforward in a linear line. But the truth is, for those of you who have done or are doing a thesis, it's more like this, right? So let me start with giving you some forward-looking statements. All opinions that I will express over the next 20 minutes here are based on my own experience. All career decisions I have taken were of personal nature, and I didn't take them on my own, but together with my wife of 30 years almost, which I consider as my biggest achievement so far. So first advice to you guys, find your significant other in whatever size, shape, or form you prefer. Third, I urge you not to copy what I'm going to tell you. This may not work for you. It worked for me. It may end in disaster for you. So please don't copy. My way, and that's the structured CV I was referring to. I did pharmaceutical studies at the University of Frankfurt in Germany. Why, I come back later. Then I went on to do a Ph.D. Because I had very bad lab assistants during my studies of pharmaceutical sciences. So I said, I can do this better. That's why I started my Ph.D. Did my Ph.D. at Frankfurt University as well at the University of Kansas in the United States. And in between, I was working in the pharmacy night shifts or during the weekend in order to earn some more money. And I applied and got a postdoc position at the University of Leiden, the Leiden Amsterdam Center for Drug Research in Holland. Stayed there for 15 months and then was hired as a senior scientist or MER at the University of the Saarland in Germany. And I stayed there for something like 2.5 years until I moved back to the University of Leiden as a tenure track assistant professor in pharmaceutical technology. I obtained my tenure after only 4 years, and I became an associate professor in 2002 at this very institution. At that time, my head of department left, so I told my scientific director that I could take over the job of the head of department. They declined because they wanted somebody from the outside to, bring in new impulses or whatever. So I got a little frustrated, and I looked around a little bit. I got an offer by industry, by Enzon Pharmaceuticals, a biotech company in New Jersey, who hired me as their vice president of research, where I was responsible for early pipeline projects. And now, all of a sudden, I was the boss of 40 people in my research department. Then I went to a scientific conference, where a former professor of the University of Geneva gave a talk. 300 people in the room. I stood up and asked a question, as you do. So later on, Robert came to me and said, we talked about his presentation, and then he mentioned that there was a position open at the University of Geneva in Biopharmaceutical Sciences. I applied, I got the job. And since then, I'm stuck here in Geneva since 2005. And I had the honor to be an invited professor in 2015 at the University of Graz, one of the most beautiful towns in Austria. And so you see, I worked in 5 countries in different roles. I am fluent in 4 languages. I've been around the block. Talking about biopharmaceutical sciences. Now, this always fascinated me from the beginning of my studies, this interaction of drugs or drug formulations, drug carrier systems with biological systems. Now, what are these biological systems? Well, starting from Leonardo da Vinci, we looked at the body, then we got more and more complex. We invented the microscope to look at tissues. We look at single cells. We look at protein networks, how different mechanisms influence each other in pathology or in the healthy state. Now we can even look at single molecule and protein interactions, like this receptor interactions. And of course, that on the DNA and right now on the RNA level, we have information on how this works, how these biological systems work as well. And based on this information, we were able to develop medicines and treatments. However, as I mentioned, this became more and more complex, and this knowledge of biological systems became more and more complex. Now, I think we can all agree that nature is complex indeed. Here you see an image of the activation of immune cells in order to attack cancer cells. And this is, by the way, the project, the Ph.D. project of one of our hosts, Maria here. And she is only looking at this small interaction here, the STING pathway, where you add some cGAMP ligands, and this is then activating these immune cells in order to attack cancer. So indeed, this is really complex, and we can look with our projects only at like tiny parts of it. But we have to understand the whole of it. So what? Well, diseases are complex, and they're dynamic as well. Here you see a image from a bioinformatics analysis. Each of these points are giving you an information about different proteins, different mechanisms, different enzymes, receptors that are involved in a disease, from the baseline on the treatment after 1 month, 3 months, and after 1 year. And you see, this is not only getting less, but there are different mechanisms, different proteins, different receptors involved in the whole disease development and treatment development. So these diseases are not only complex, they are also dynamic over time. And what making it even more complicated is that these diseases are really personal. You can't treat a certain disease for the big majority of people by just using one treatment, one dose, one drug, or combination of drugs. However, there are always outliers, like here, or like this Dutch lady here on her bike, which need more precise treatment. So the treatment or the understanding of diseases and their treatment must be precise, because diseases are rather personal, right? Yes. If all the tools you have is a hammer, all problems tend to be nails. But of course, you cannot, screw in a screw using a hammer. So what is needed is an approach that takes into account this dynamic complexity of diseases and the dynamic complexity of treatments. And how? Well, we have the answer because we are here in Switzerland. We know all about complex tools, right? So one of these complex tools is nanomedicine, or nanomedicines, if you want. It's just a part of the pathology-based approach, how to treat diseases or manage diseases today. Here you see a very nice image from drug discovery today. The break-in of paradigm, so the paradigm shifts that occurred over time. From the early 1900s, maybe, we had observational treatment, herbal medicines, and so forth. Then we had a paradigm shift into the chemistry and biology-based medicine. We looked at synthetic drugs, receptor theory, which was formulated already earlier than 1950, however, now became all of a sudden very important. I saw the advent of pharmacokinetics and so forth. And all the chemistry drugs were developed like penicillin, propranolol, cimetidine, and so forth. And we had another paradigm shift, and we looked more at the pathology. So today we're at the pathology-directed medicine, including human genetics, molecular biology, systems biology, and for us pharmacists, systems pharmacology, how to take into account this complexity of diseases and their treatment. And that has resulted in therapies like these hit them hard, hit them early, heart, HIV triple therapy, antibodies, antibody drug conjugates, gene therapies, ATMPs, and so forth. Now, in my group, we're working on nanomedicines and the characterization of which and the design of which and so forth. And one of the aspects that is concerning our work has to do something with Shakespeare's Hamlet, because there he says, we know not what we are, we know what we are, but not what we may be. Let's let this sink in for a while, but what it means is actually this. When we prepare our nanomedicines in the lab here, we can do the characterization of the synthetic identity of these particles as liposomes. So we have an understanding of the size, of the chemistry, and the shape. Once you introduce that into a biological system, the nanoparticle would take on a biological identity, which means you will have a difference in size. You will have adsorption of blood components to the surface of these particles, which is then called, corona around the particles. You may have aggregation. Now, the physiological response in the end depends on not the synthetic identity, but on the biological identity, because this is what the body sees, the immune system sees, the receptors see, and so forth. So our interest now is, can you design the biological identity of a nanoparticle by choosing certain physical-chemical properties? You influence the biological identity, which then determines the physiological response. This is one of the questions we have currently. So the output so far from my career is more than 150 publications, approximately 11,000 citations with an H factor of 52. H factor is also age-related, of course. And of course, I did not write all these publications myself. Most of it was written by the people I had the pleasure and the honor to work with. 22 book chapters, co-edited 2 books with friends of mine, and I'm named as an inventor on 10 patents. So this is the straightforward CV-related information I wanted to give you. Oh, and I have also 220 invited talks and so forth, including this one. But how did I get here? And I call this the red and the green. You know, at the University of Leiden, they sent the young professors to seminars to learn about leadership, to learn about how to do their job. And there I learned that there's always a red organization and a green organization. The red organization is the rules, how you have to do it, how it's written down, the règlement. The green organization is how you actually do it. Give you an example. I had to install at the University of the Saarland, I had to install a first aid kit in my lab. So I called or sent an email to the people who are responsible of putting this first aid kit on the wall. I never heard something back from them. So that was the red organization. You tell them, do this, and they do this. They didn't. So I took a drill and I put this thing on the wall myself. This is the green organization, just for give you an example. Now, coming to the green background of my CV, I was 7 years old. I read a comic strip, which was very famous at the time in Germany, Fix und Foxy. And there he was, Professor Knox, who was standing in front of a blackboard. And on this blackboard, there were equations, there were chemical structures. I didn't understand at the time, of course. And I told myself, Gerrit, if you grow up, you will understand all of this. You will become a professor as well. And then at 13 years old, I was brought up as a Protestant. So at 13 years old, I got my confirmation, and my godfather, a teacher, gifted me with the best present ever. He gifted me these two books that I still have here in my library, one on inorganic chemistry and one on organic chemistry. That's really gave the initiation for me to be very much interested in science as such, and also in chemistry and everything that is related to it. 19 years old, I graduated from high school, and instead of going to the army, I didn't want to become a soldier. I did my civil service, and I worked with old people, handicapped people, and terminally ill people. Basically, I accompanied them until their death, and that influenced me and impacted me in a way that I said, okay, I can maybe combine my scientific interest that I already had with some medical aspects. So, I mean, the decision was really clear. Pharmaceutical Sciences combine both. Plus, there was a 70% female students there. So what could you lose? So I decided to study Pharmaceutical Sciences. And something happened, 1989. The Bangles were on with Eternal Flame, were in the charts. Indiana Jones was on his last crusade, and there was something happening with a wall somewhere in Berlin. I was still at Frankfurt, just graduated from my studies. And I talked to one of my professors, pharmaceutical technology professor, Jörg Kreuter, asked him whether I could do a thesis with him, because pharmaceutical technology is pretty easy, right? So he threw expressions at me that I never heard before. Nanoparticles, complement, drug targeting, reticuloendothelial system. So I scratched my head and I went into a library in order to look up these expressions. Our library, this is sort of like an offline Wikipedia at the time that we had with books. Anyway, so I decided to do this. A couple of years later, I became assistant professor, associate professor, and 39 years old, I was invited to AstraZeneca to interview for a job in industry. And I was, let's say, underwhelmed by the offer they made me in terms of the task I would have. So I sat at the airport of Malmö at 39 years old in the middle of winter, and I asked myself, what am I going to do with the rest of my life, with the rest of my career? Do I want to stay in Leiden? They don't want to make me boss there. So what am I going to do? So I never thought I would, in a presentation, I would cite the Bible. But this is all about finding your passion. If I have the gift of prophecy and can fathom all mysteries and all knowledge, and if I have a faith that can move mountains, but I do not have love, I am nothing. That means only what you love to do, what you are really able to do, what you have fun doing, you can be good at. So the basic question there at this airport in Malmö was, what do I love doing? 3 things. And I advise you to do the same and find your passion. First of all, I'm a teller of stories. I can present, give me a topic and a microphone, and I can entertain you for half an hour. And this is really, really important, as we learned, my wife and I, when we went to Australia a couple of years ago. We learned that the Aborigine culture is actually based on telling the stories from the grandfathers to the grandchildren. And they preserved these stories exactly over 50,000 years. Astronomy, where to find water, and so forth. They preserved this over this long time. So never underestimate the power of a good story. Secondly, I like to be exposed to new stuff, learn a new language, work in a different environment, travel. And I do this by jumping right in, without ever knowing when I reach down there, whether I can swim or whether I'm far from the shallow. Sorry. Third thing I like to do, and what I can do, I can put together teams to work together and walk in the same direction. At the University of Leiden, as assistant professor, I had a Ph.D. student, male, very hierarchical, macho from Iran, and the same time, lady from Israel, who used to be a sergeant in the Israeli Defense Force. They did not get to love each other, but they respected each other and they worked together. And that's quite an achievement. However, what I also learned, and I will come back to this in a minute, the first rule of leadership, if you are now in the situation where you graduate from your PhD ship, you did your postdoc, you found a job in industry, the first rule of leadership, and you have to lead in a lab or a couple of people, the first rule of leadership is everything is your fault. If it goes wrong, it's your fault. If you succeed, it's the team's effort or individuals in your team. Now, let me finish with a couple of pearls of wisdom. Leadership is given directions, even though you are lost yourself. It's taken decisions in the absence of sufficient information. And those of you who have served in the army as officers, maybe, you know that. Always remember that people are different. People are not their resume, as I tried to point out. There's much more behind this. If you succeed, as I said, it's your team. If you fail, you are the only one to blame. And always hire people that are, or at least have the potential to be better than you. Saves you a lot of time, saves you a lot of work. And trust me, it's much more fun to promote people than to fire them. Trust me, I know, I've done that. Last but not least, responsibility and empowerment go hand in hand. If you want something done by a member of your team, you only do not have to give them responsibility, but also the tools to achieve this. Don't ask what the boss thinks. Tell the boss what he should think. Nobody sees you coming in at 6 a.m. Everybody sees you leaving by 5 p.m. That's especially a problem that I have because I'm very early in the office. And I believe that if you work more than 10 hours a day, you are not very productive. You have a management problem. You have a time management problem. With this, I'd like to finish. Would I do this all over again? Yes. Would I do this today? I don't know, because today we have a lot of problems. We have a pandemic we have to live through, and this will not be the last pandemic. We have an ecological crisis, a catastrophe in the making. We have economical crisis and so forth. But remember, whatever you do, I was 2 years ago, I was running in these beautiful mountains in Alaska. There were bears in the area, as the rangers also told me. And it was raining all the time, all the 2 hours I was there on these mountains. However, that's not a reason not to have fun. So with this, I'd like to ask you, whatever you do, have fun and good luck out there. And thank you very much for your kind attention.
Wow. Thank you, Gerrit. This is a fantastic presentation, and I have definitely been inspired with pearls of wisdom. We have questions from the audience, and we have time for one quick question. So I'd like to pose this to you. So, how did you reconcile personal life and professional life while having all these worldwide opportunities? And this was asked by Anne Nguyen.
Yeah, that's a very good question, Anne. Well, first of all, I have the privilege to be married to a very forgiving wife. Secondly, we always understood that we have to take any career decision together. So if she would have not agreed to go to New Jersey, we would not have gone there. So I think this is one of the major things you need to understand in a partnership. Secondly, we do not have kids, which made things a little more easier. I have a postdoc that currently recently moved to Oxford from Geneva with 2 little kids, and that's much more complicated. I admit this.
Yeah, I can imagine. Thank you, Gerrit. Thank you very much. And thank you for the questions, audience. Do you have any final comments for the audience to wrap up?
No, I think I did say everything I wanted to say.
Nice.
Will the questions stay in the chat there so I can answer them?
We will have the speaker sessions this afternoon from 1 to 2, which is specifically for this. So again, all the participants, please, I remind you that at 1 p.m. we have the speaker sessions, and you can ask any speaker all the questions you like. So we hope to see you then again, Gerrit.
Okay, fair enough. Thank you.
Thank you very much. All right. So I hope you were inspired as I am. And without any further hesitation, we will move on to the next speaker. And I remind everyone, please, questions, put them in the Q&A at the right-hand part of your screen.